Meiosis. Now this deals with sex cells, all right, or gamete cells. They're the same thing. So let's compare and contrast the body cells that we've looked at in the previous video with mitosis and the gamete cells or the sex cells that are required, that we need to look at for meiosis. So in somatic cells, all right, these are the body type cells. They undergo mitosis. For what? To grow, repair damaged cells, or replace dead cells. And they're also known as diploid cells. So why diploid? Di stands for 2, right? And we're going to use this as a coefficient for a variable, n. So for every value that this represents, it's going to be doubled. So if n here represents particularly regarding the chromosomes that are in each of these somatic cells, what do you end up getting? You get 46. So you have 46 that are the total. So when we make these daughter cells, and each cell has 46 chromosomes, and every single cell is genetically identical to one another, that is representative of a body cell. In contrast, however, gamete cells, or sex cells, undergo meiosis, which is the focus for today's video. And this one is to create genetic diversity. So make genetic, look at this, genetic diversity. And it's also to reduce, I'll just put here, to decrease or reduce the chromosome number from diploid to haploid. So if our diploid was 2n, we're going to change it to just n. We can't make this n value representative of pairs of chromosomes because pairs are always in 2, which is what a diploid is. If we don't have that 2, this is not a pair, so this is just 23 chromosomes. All right, so then this will be the total. So 23 chromosomes in total. Because we only have half the number of chromosomes, where are the other half of chromosomes going to be able to come from? Well, let's say this is one parent. Obviously, the other half, the other 23 chromosomes, needs to come from the other parent. So that when you combine them together, you get a diploid again. So when they do get together, you now have a daughter cell that is genetically unique, very different to its former parent, because this time the baby is going to have half the chromosomes from the mom and half the chromosomes from the dad. And if you're probably thinking, why must these gametes be haploids? Okay, why do they have to be N before fertilization? Because honestly, the haploids can form a diploid zygote, but we want them in equal amounts of genetic material from each parent. The more genetic diversity it has, the better, as this can increase resilience. So now let's assume they've gotten together, right? So let's say this is the egg here. So this is one gamete cell. And let's say this is the sperm here, the other gamete cell. They get together. You now form that zygote, right? So that zygote, once fertilization happens, eventually becomes a blastocyte. So if the egg was just 1N and the sperm was also just 1N, when you put them together, you get 2N for the zygote. Again, it is a diploid now. This was a haploid. This was a haploid, right? Now what happens to the next step in the blastocyte? Well, you have an inner cell mass over here. Why is this significant? Because this is where differentiation starts to occur. Differentiation is when there's cells that specialize to become a certain type of cell. So at this point, we have these cells that are non-differentiated, so they're like stem cells that aren't a particular type of cell, but at this point they start differentiating. So some of these stem cells start to become maybe blood cells, all right, and then maybe other stem cells, they start to become, skin cells, and then you have another stem cell that start to differentiate and specialize into becoming a nerve cell. Okay, this is where the differentiation occurs. And then we get to the gastrula. Okay, when we get to the gastrula, it becomes even more complex. All right, so again, at the blastocyte, this is where cell differentiation occurs. Now the gastrula gets even more complex because at this point over here, the ectoderm, the mesoderm, and the endoderm starts to form. Why is this important? Because this gets even more grand. The ectoderm forms the external structures, for example, like the skin, right? They're made up of epithelial cells, and I believe there may be even the nervous system as well. But when we talk about the mesoderm, they start to form and drive the development of structural support, like transport systems, okay? So musculoskeletal, for example, or the cardiovascular system. Maybe even excretory would fall under that too, because that's also a transport system, right? So this type of derm is for that, is for those kind of functions. And then the endoderm creates more of the systems that are of the internal effect or the internal linings. So obviously like the digestive system is one, which obviously hosts like the gastrointestinal tract, the liver, the pancreas, accessory organs, etc. And then it starts developing into the embryo next. And the embryo is like the beginning stages of finally that fetus, okay, of fetal development. And then the fetus continues to grow and grow and grow and is now very, very close to being birthed out and continue to grow into a full-fledged human being outside of the womb. All right, so meiosis has 2 rounds of division, okay, of cell division. All right, the first one is known as meiosis 1. And this is very, very similar to what we looked at in mitosis, okay? There is a prophase. There is a metaphase. There's also an anaphase. And there's a telophase. However, all of these are going to be identified with a 1 in front of them because, obviously, we have 2 rounds of it. So the next set is going to be prophase 2, metaphase 2, etc., etc., okay? And this is all going to be concluded with cytokinesis. Once that's concluded, we can now move on to meiosis 2. Meiosis 2, it's the same thing, all right? Prophase 2, and then metaphase 2, and then anaphase 2, and telophase 2. And this is also concluded with its own cytokinesis. And this is the very, very final step. Now, if we look at 2 types of chromosomes, and let's assume that they're the same size, they are now matching, all right? We call these homologous chromosomes. All right, matching. All right, so they have the same size, obviously, but then they also have the same type of genes. All right, so obviously in the same locations, right? Remember we talked about loci in the previous video? So obviously they have to have same type of genes because if this is about eye color, then this one needs to be of eye color as well because this is the parent. Because this is one parent and this is the other parent. So to ensure that there's genetic variation, particularly about eye color, both of them need to have eye color so that they can mix and match and see eventually what's going to happen with their kid. Because the kid could either have one eye color from maybe this parent or it could have the eye color of the other parent. So it's very important that this matches. Okay, so this is also known as a tetrad, okay? So it's a pair of these homologous chromosomes, okay? This is a homologous chromosome, this is a homologous chromosome to each other. All right, so this is known as tetrad. Obviously, tetra meaning 4 if you've taken chemistry. So obviously this here is one chromosome, this here is the other. Okay, they just replicated, right? But they are each a chromosome. So this is 1, this is 2, and same thing here. This is 3 and this is 4. Now, in order for meiosis to start, it needs to have interphase kick in first. Because this is the period between the cell divisions. Because as illustrated in the previous video, this is the phase where there is primary growth. Okay, remember the G1 phase? This one was the primary growth. So primary growth phase. We then end up having the synthesis, right? So synthesis is the replication. okay, of the chromatin. Remember what I just showed you? This chromatin is going to replicate. Now you have two chromatin. Okay, so this is a chromosome now. Same thing for maybe the other parent. Okay, this was one chromatin. It's going to replicate to make the other chromatin. Okay, there is replication of that chromatin. And then we have the G2 phase. This is when the organelles replicate. Okay, very similar to what we just looked at in the previous video of mitosis. All right, so before replication, I want you to think about this. How many chromosomes are there? Well, if you want to get very, very technical and only talking about one parent, so let's talk about the green one over here. Let's say this is the first parent that we're observing. This is one chromatin, right? But how many chromatins are there in each of our cells during interphase? We have 46, all right? So you're going to have like 46 of these, all right? All right, so there's going to be 46. So particularly 46 chromosomes. But because this is not a fully fledged chromosome where it looks like this that we're used to seeing after it's replicated, this one can also be known as 46 chromatin. All right, so again, this one times 46 of them. All right, but how about after replication? After replication, so let's have that chromatin again. It has replicated, okay? And remember, there's still 46 of them, right? This is obviously a chromosome. So we still have 46 chromosomes though. It seems like nothing has really changed. But how many chromatins are there? Okay, because at the beginning, this here was a chromatin. After replicated, this here is a identical version of that chromatin. So technically, we actually have 92 chromatins. Again, if you looked at my previous video for the mitosis and heredity, the vocabulary jargon is very, very annoying. Some individuals might consider this 46 chromatid, okay? This one too, 46 chromatid with a suffix of TID. Some people only use those when we use the word sister chromatids. So this here may be a sister chromatid to this sister chromatid. Whereas this one, because there's no sister there, it's just called a chromatin. However, some individuals could deduce this as a chromatid as well. But again, it's just semantics over there, okay? The important aspect that I want you to understand is that the chromatin or chromatid number is going to be doubled compared to the original number at the very beginning before replication. All right, so now let's get into prophase 1. Let's start meiosis now that we're ready because we've replicated. So now that we've started with interphase, we're now ready to get into the actual division section of meiosis. So let's start with prophase 1. So let's get a big chromosome over here, but there needs to be a homologous chromosome that is of the same size right up to that one. So let's put that down here. And then let's get another chromosome, okay? But this one might be a little bit smaller. So that's going to have a homologous chromosome as well. Okay, so this one is kind of the same size as that now. So remember, 2n is a diploid. We are still a diploid because we haven't separated in half to create the daughter cells for meiosis. Okay, we're still at the beginning stage. So we're still diploid right now, which means we have, all right, so let's start off with prophase 1. Okay, let's have this chromosome the same size as maybe this chromosome over there. And then maybe I'll have a smaller chromosome over there, which is going to be a homologous chromosome to this chromosome over there. So obviously this and this are going to pair up, whereas this one and this one are going to pair up. And I know it looks like there's only 2 pairs, right? However, in every single one of our cells, there are 23 pairs. So there should be 23 pairs in here, also known as 46 of these X's, right? These chromosomes, but obviously that's going to be impossible to fit. So we'll just use 2s, okay? 2 pairs, so 4 chromosomes. So like in mitosis, centrioles start to move to opposite poles. All right, which means the SF, right, the spindle fibers, form from there. Remember what these spindle fibers do, right? They start attaching to the centromeres in the middle of these chromosomes, right? And then it pulls them apart. And you notice how the chromatins has condensed quite well to make it into a X-like shape of a chromosome, a fully fledged chromosome, all right? So it's already done that, as you can see there. It's not like strings of spaghetti anymore. So again, chromatin. condensed into chromosomes, and the nuclear membrane is no longer there, okay, or it's starting to be no longer there. So maybe in order to make it more accurate, I can kind of draw a little bit of that nuclear envelope, all right, but technically it is pretty much gone, okay? Nuclear membrane, nuclear envelope are synonymous. They're the same thing. A very important thing for you to note here, I'm going to be putting a star here, is that there is some crossing over that occurs. So while this homologous chromosome pairs up with that homologous chromosome, genetic information is exchanged, forming something called a recombinant chromosome. Creates recombinant chromosome. We tend to use the word recombinant for like genetic modification, whether that's gene therapy or something that you're more aware of, like genetically modified organisms like GMOs. Hence why it's kind of used in this sense as well. We are putting some genetic variants into these chromosomes when they start becoming different types of sex cells, okay? Once we finish this whole process of meiosis. So we're creating something new, genetically modifying it. What happens next is metaphase 1. So this chromosome is now paired right beside this homologous chromosome, and this smaller chromosome is now with the other smaller chromosome. And remember, we have some centrioles there, so this attaches there, this attaches there, this attaches there, this attaches there to those centrosomes, right? Like over there, in the middle, that is connecting the sister chromatids together within that chromosome. But remember, they did hold hands at one point in prophase 1, right? Just like how they're holding hands over here. So they are actually intertwining their genetic information. And again, this guy and this guy are going to be holding hands too, okay? And crossing over. That's what you kind of see here. Okay, it doesn't look like they're holding hands, so maybe I'll just put them a little bit closer. All right, holding hands, holding hands. Okay, so now they're both very clearly holding hands with one another, and they're exchanging the genetic information since this phase up here. They're just lined up really well at this point. So by the time we split them up to this side and this side, you're going to have a mix of colors. So this side is going to have some white and blues, this side is going to have some white and blues as well. All right, so like I've already said, the chromosomes lined up at the equator. All right, with their homologous pairs. So both of these tetrads have lined up perfectly. And then we'll get to anaphase 1. All right, as you can see, it is very, very clear that they have exchanged genetic information extremely well and intertwined. Okay, again, starting from prophase 1, they've already started doing that. And by metaphase, there is full genetic variation. Okay, so now when you pull them apart, you're moving them to opposite poles now. All right, so then these chromosome pairs are pulled to their poles, all right, by the shortening of those spindle fibers. And the important thing that I want you to know here is that the sister chromatids are still intact. All right, so we're not separated. Okay, this is different than mitosis, all right? In mitosis, if you had maybe a chromosome here, if it got pushed in opposite directions, this actual centromere got split apart, resulting in it being pulled away like that, and this one being pulled away like that. It's literally cut in half. That is not what we see happening in anaphase 1 of meiosis. All right, so only the tetrads are broken up. Remember, this was one chromatin, this was another chromatin, this was another chromatin, this was another chromatin, all right? So this tetrad just exchanged genetic information and just got pulled apart, but did not actually separate in half. And then we eventually get telophase 1. And you notice how the nuclear envelopes have started to come back, all right? So nuclear envelopes, or Ne, nuclear envelopes, start to reappear. All right, but now there are two of them. Two of them. And at this point, a cleavage furrow starts to concave inwards again. within that cytoplasm so that it can eventually split in half, all right? All right, forms. Getting ready to pinch this. All right, so let's assume they've broken in half. We now have one cell there and another cell there. The nuclear envelope should be completely evident by now, and those chromosomes are now comfortably sitting in each of those. So again, this is now cytokinesis. And remember, this is just round one, all right? Telophase 1, anaphase 1, metaphase 1, prophase 1. These are all just the first round of meiosis. Now we got to do a second round, all right? So this is an official daughter cell, genetically different. This one here is, again, another daughter cell, genetically different. And each daughter cell here has half the number of chromosomes as the parent cell. Remember, the parent cell had 2n, okay? 46, right? 23 times to give you that 46, so there was 46 at the beginning. Right now, the pairs are gone, right? So let's look at this pair, and look at that pair. They're homologous to one another, right? But they're separate from each other now. So you can't do 2n over here, because even though we have 23 chromosomes in here, I can't multiply that by 2, because there are no pairs, right? This and this is not together. This and this is not together. They were the pairs. So because they are no longer together, I cannot put a 2 in front as a coefficient. So it can't be 23 times 2. We literally only have 23 chromosomes. So this is half and this is half. Alright, so we are no longer diploid, we are haploid. Alright, so I'll put n is equal to 23 over here, n is equal to 23 over here. Okay, this is a haploid. This is also a haploid. Alright, let's go to meiosis 2 now. So it's very similar to mitosis, but there is no duplication of chromosomes. Okay, remember we had interphase at the very beginning of meiosis 1, same as mitosis. In meiosis 2, there is barely an interphase. Alright, so some textbooks actually say there is no interphase at all. It just goes straight into prophase 2, okay? But technically, there is still an interphase just before the prophase 2, okay, during this meiosis 2 cycle. It's known as interkinesis. So everything happens as normal, just like any other interphase, right, from meiosis 1 and mitosis. But the only difference is no replication. All right, that's the important part. There is no replication in this very, very brief interphase period, okay, interkinesis. All right, so this one kind of looks like what we just did, right? Cytokinesis. But as you can see, the nuclear envelopes here are starting to fade out. Okay, they're disappearing. So obviously this is not cytokinesis. We have now entered meiosis 2 very evidently because those are going away, okay? So this is obviously prophase 2 now. Okay, so we've gone through that interkinesis where there's a little bit of resting time and more growing, but again, no replication, and now we're going straight into splitting these two apart and these two apart as well. So you're probably wondering, are we going to be able to get 4 cells? Okay, 1 cell, 2 cell, 3 cell, 4 cell? Okay, when this and this split off in half? Yes, that's what we're going to be doing. Okay, so then there's some centrioles here, centrioles, right? And then they moved to this pole, right? This also moved to this pole, this one moved, and this one moved to their poles, okay, on opposite sides, getting ready to pull them apart, just like how they've always done in the previous stages of prophase. All right, again, nuclear envelope breaks apart, and then the SF, right, the spindle fibers start to form again, then the centrioles move to the poles. In metaphase 2, they do not line up beside each other like this, because obviously they're not homologous. They can't line up beside each other. They don't match, so they line up on top of each other, all right? But also the chromosome pairs are at the equator again. And those spindle fibers attach, like always, to the centromeres of those chromosomes. But again, the important thing to note is that these sister chromatids, okay, within this chromosome, same as these ones here, here, they are no longer identical because they've crossed over. All right, so that's the big difference. So when they start to get pulled apart this time, they are genetically different now. All right, so now they're being pulled apart towards the opposite poles, and the chromatids, all right, the sister chromatids are now being completely ripped apart in half. So the spindle fibers pull sister chromatids apart. And like usual, the nuclear envelope reappears. Right, and now they're becoming spaghetti-like again, right, these chromosomes? So they uncoil. And of course, the spindle fibers are gone. And then of course, there's going to be some cleavage furrow here. It's going to split apart in half. Same thing with this one, split off in half. So now we have 1, 2, 3, and 4 daughter cells. But each of these 4 genetically different daughter cells, but each of these daughter cells that are now present are genetically different. All right, so each daughter cell here still has half the number of chromosomes as the diploid. All right, again, this is cytokinesis, right? This is the very, very last step of this phase of meiosis. All right, so remember, before meiosis 2, we had 23 chromosomes, right? It was a haploid, remember? But remember, the chromosomes look like this, like an X-shaped structure. Okay, well, one was big, right, and the other one was small, and then they were already intertwined with genetic variation. But how many chromatins are there? Remember, this was 23. Okay, we see 2 here, but technically there's 23. But remember, this is a sister chromatid to this one. All right, so this is one chromatin, this is another chromatin, so it doubled. Okay, same thing with this one here. This is one chromatin, this is another chromatin. So technically, even though we have 23 chromosomes, we actually have 46 chromatids. But after meiosis 2, right, as you can see up here, they're not in an X-like shape, right? They're more like that and that, obviously intertwined with genetic variation already. So still, again, I know we see 2 over here, but technically we assumed that the 2 chromosomes we drew represented the 23 chromosomes. We just can't fit 23, right? So this is also 23 chromosomes. Remember, we have to call this a chromosome because, as exemplified in the previous video, when they do get split apart, they are no longer called chromatids, okay? They're called official chromosomes, even though they don't look like an X-like shape, right? But how many chromatins are there? Well, technically it already is, because this one doesn't have its replicated chromatin anymore. Okay, it's all on its own. Same thing with this one. So unlike this one that has 2 of its chromatins, same with this one, 2 of its chromatins, this one does not. So we can't duplicate it. We can't times 2 like we did up here. So this 23 stays as a 23. So now we have 4 haploid gamete cells or sex cells. And there are different combinations of alleles for different genes along the chromosomes. Now errors do occur, particularly something called non-disjunction is the failure of those homologous chromosomes to separate properly. And maybe not even the whole chromosome, maybe even the sister chromatids of that chromosome, they also don't separate properly. All right, so it's the failure of either the homologous chromosomes or the sister chromatids, okay, that are attached to that centromere in the middle to separate properly. Okay, it can happen during meiosis 1 or meiosis 2 actually. So let's look at the case of meiosis 1. Metaphase 1, when we had this bigger sized version of these homologous chromosomes lined up together and these smaller ones lined up together, and they lined up side by side so that we can just split them apart and not cut their sister chromatids in half like that or like that. Instead, we just pull them apart to this direction and this direction. Well, that gets to anaphase 1, right? So in anaphase 1, obviously these 2 should go in that direction and these 2 should go in that direction. In anaphase 1, if there's a mistake, sometimes you end up getting this. Notice what happened here, right? It's supposed to be over here, right? Because that was this chromosome should be on that same side. So if it started on the right side, it should be ending on the right side here, but now it's on the left side, right? This is a case of non-disjunction in meiosis 1. And then by the time you get to metaphase 2, okay, so now in meiosis 2, if this were to have continued, this is what you would see. And then of course, when we get to anaphase 2, this has got to split apart and literally break these sister chromatids in half. And same thing with this side. And this chromosome too. It's got to split that sister chromatid in half in one direction and the other direction too. As you can see, they're separating away right now. And then the next phase will be the splitting of this. Alright, starting in telophase 2, and then eventually cytokinesis splits them apart completely. So you can see where I'm going with this. And by the very end, you're going to have still 4 cells when they all split apart, but rather than this one having 2 chromosomes, which we represented as 23, right? We assume that there will be 23 in there. You would have a +1 over here. So it'll be a 24th. Same thing with this one. This one over here, again, was supposed to have 2 chromosomes, okay, over in there, representing as 23 chromosomes. But because we only have 1 here instead of 23, we got a -1, so this would be a 22. Okay, so we only got 22 chromosomes in here. Same thing with this one. So we have a normal number of 23, but +1. Same thing over here, 23 plus 1. This one had a normal amount of 23, but you had to subtract 1. This one also 23, subtract 1. These are known as trisomy. These are known as monosomy. So that trisomy is just that one extra chromosome in humans, right? So when a zygote does form, it will have 24 pairs of chromosomes instead of 23 pairs. For monosomy, it's missing 1 chromosome, so when the zygote forms, it will only have 22 pairs of chromosomes instead of the 23. Now how about in meiosis 2? Let's look at this case. Everything goes well until metaphase 2 and anaphase 2. So let's look at metaphase 2, how they aligned. But in the next step, when they try to separate towards their opposite poles, the splitting of the actual sister chromatids, so from here and here and here and here, had an error. Let's have a look. You notice what happened over here? This was supposed to cut in half into their respective sister chromatids, but it didn't, and the whole thing went towards the left, which ended up like that. This side seemed to have done it fine, so there's no problem there. It's just this one. So if we were to keep going and finish off with telophase 2 and cytokinesis 2, how is this going to end up? Well, the cleavage furrow here starts to completely break in half, so the cytoplasm completely concaves inward and splits apart. Now we have those 4 daughter cells again, that's no problem, but this one has an extra amount, right? So if this was 1 chromosome, half of this one would have been the second one, right? So then 2 of those are considered 23 chromosomes, right, in our mind. But because look over here, what about the other half that we haven't taken into account for? Okay, it's still sticking there. That counts as an extra one. So it'll be a 23 plus 1. So this would also be trisomy. How about this one? There's only 1 chromosome. Normally we're supposed to have 2, which is 23, right? But because we only have 1, instead of a 23, it'll be -1, so it'll be 22. So this will be monosomy. This one has 2, 1, 2, which will be represented as 23 in the back of our head. So this will be 23. So this will be normal. This will be no issue. Same thing here, no issue. A well-known example of monosomy is Turner syndrome. A well-known example of trisomy is Down syndrome.